Hello, my name is Isabelle Olejniczak, and I am a senior in Health Sciences Pre-Medicine. Today I'm going to discuss my project from this year, the association with glutamate and glutamine levels in periaqueductal gray with neuropathic pain in IBS patients. To start off, I would like to share some background information that is crucial to understand the study. As mentioned before, we are looking for a correlation between pain and neurochemical levels in patients with irritable bowel syndrome. Pain is an unpleasant sensory and emotional experience that may be associated with tissue damage in any peripheral part of the body. There are three categories of chronic pain. This would include nociceptive or inflammatory pain, idiopathic pain or pain without an origin, or the pain specifically targeted in this study, which is neuropathic pain from nerve injury. This form of chronic pain is experienced by those with irritable bowel syndrome, or IBS for short. IBS is a chronic disorder that affects primarily the large intestine and causes gastrointestinal symptoms such as abdominal pain, diarrhea, bloating, and gas. These patients experience hypersensitivity and pain when bloated due to abnormalities in endogenous pain inhibitory mechanisms. The midbrain periaqueductal gray, or PAG for short, which is shown in Figure 2, is thought to serve as a control center for pain and may play a role in hypersensitivity experienced by IBS patients. This is because the PAG plays an important role in the pathway of pain. It starts with afferent or incoming nociceptive information entering the brain from the spinal cord. The ascending information activates the neurons of the rostroventral medulla and the midbrain PAG as shown in Figure 1. The PAG is a key component of the descending pain modulatory system, or the DPMS, which controls neuropathic pain in the body. It has the inhibitory, known as GABAergic, and excitatory, glutamatergic, output to the dorsal horn of the spinal cord. In application, some fear responses induced by the PDAG are mediated by the GABAergic system, while some pain responses are mediated by the opioidergic system. Two important neurochemicals of interest in the study are glutamate and GABA. Glutamate is the principal excitatory neurotransmitter in the brain, while GABA is the main inhibitory neurotransmitter in the brain. The hyperexcitability of this neurotransmitter system may lead to the development of sensory symptoms that are observed from pain. This brings me to the goal of the study, which is to quantify neurochemicals in the periaqueductal gray from irritable bowel syndrome patients to identify the chemical differences associated with pain. The study was conducted with 25 experimental or IBS patients and 13 control or healthy subjects. MR spectroscopy was performed in the PAG with a modified semi-laser sequence. These spectra were then processed using MATLAB and later quantified with the LC model with a water scaling option using a simulated basis spectrum. The neuropathic pain was assessed using a pain questionnaire called Pain Detect on a scale of 1 to 10. The concentrations of metabolites were then determined after correcting for relaxation times, tissue water content, and CSF contributions. So as you can see, Figure 3 shows the MR spectrum for a healthy patient, and Figure 4 shows the spectrum for an IBS patient. Spectra with a good signal-to-noise ratio and spectral resolution were obtained consistently from both groups. After statistical analysis, significant differences were found between the IBS and control groups for glutamate and glutamine with a p-value of 0.03, aspartate with a p-value of 0.036, and myo-inositol with a p-value of 0.07, as shown in Figure 5. In conclusion, we have seen that IBS patients have a great increase of GLX, which is glutamine and glutamate, aspartate, and myo-inositol levels. We have not observed any correlation between neurochemical levels and the degree of neuropathic pain. Our results show that the glutamatergic system provides excitatory neurotransmission, which plays a critical role in nociceptive processing and pain modulation. The decrease in myo-inositol could be due to disruption in the cellular membrane-based second messenger system. In the future, the study can lead to the exciting possibility of developing a glutamate-enhancing PAG-targeted therapy for chronic pain conditions after seeing the results of further carefully designed studies. Thank you.